Welcome to this explainer. Imagine the microscopic world of the cell not just as a living, breathing entity, but as a dynamic, fully customizable factory. For most of human history, we were really just bystanders taking advantage of what nature already gave us. But today, we are the engineers. We actually have the tools to go in and rewrite the fundamental blueprints of life itself. It's a completely fascinating world, and I'm absolutely thrilled to be your guide through it. Okay, let's dive into this and check out our roadmap. We're going to start off by defining modern biotechnology, then jump back in time to the birth of recombinant DNA. From there, we'll meet our molecular scissors, learn how to sort things out with gel electrophoresis, and finally, we'll take a look at vectors, our delivery vehicles. All right, let's get started with Section 1, Defining Modern Biotechnology. To really wrap our heads around the sheer power of the tools we're using today, we kind of have to look back and contrast our historical approach with this new era of genetic manipulation. For literally thousands of years, traditional biotechnology was all about microbe-mediated processes. Think about your everyday normal stuff, making curds, baking a loaf of bread, or fermenting wine. We were simply utilizing existing biological processes to our advantage. But modern biotechnology, that is a massive paradigm-shifting leap. We've moved from just borrowing nature's recipes to fundamentally altering the core chemistry of genetic material. Today, we're talking about utilizing genetically modified organisms, or GMOs, performing in vitro fertilization, and creating life-saving DNA vaccines. It's a whole different ballgame. So, to capture the essence of both the old school and the new school, the European Federation of Biotechnology actually created a definition that serves as a crucial bridge between traditional uses and modern molecular engineering. They define it as the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. I know, it's a bit of a mouthful, but it perfectly sums up how we've merged natural biology with cutting-edge science. Moving right along to Section 2, the birth of recombinant DNA. Every great scientific revolution has that one inciting incident, right? Well, for modern biotechnology, that spark happened in 1972 when a scientist named Herbert Boyer took a trip to Hawaii. By 1969, Boyer had been studying these specific enzymes in E. coli that could snip DNA strands, leaving what he famously called sticky ends. Now, while at this conference in Hawaii, he happened to bump into Stanley Cohen. Cohen had been studying plasmids, which are these small, freely floating ringlets of DNA that act like microscopic delivery trucks. It was literally a match made in scientific heaven. Boyer had discovered the cutting tool, and Cohen had the delivery truck. By teaming up, they successfully constructed the very first artificial recombinant DNA molecule. It's incredible. Let's walk through the exact steps Cohen and Boyer used because it's basically a microscopic puzzle. First, they isolated an antibiotic resistance gene by cutting a piece of DNA straight out of a plasmid. Second, they used Boyer's enzymes to make precise cuts in the DNA at highly specific locations. And third, they linked that cut piece to a native plasmid vector of a bacterium called Salmonella typhimurium. To do this, they used an enzyme known as DNA ligase to act as the glue. And just like that, the entire discipline of modern biotechnology was born. Which brings us perfectly to Section 3, Meet the Molecular Scissors. So how exactly do we cut DNA with such insane precision? I mean, you obviously can't just use a tiny pair of craft scissors. The very first restriction endonuclease ever discovered was called Hind2. But wow, we have come an incredibly long way since then. Today, we know of more than 900 restriction enzymes, and we've isolated these from over 230 different strains of bacteria. Think of each one of these 900 enzymes as a highly specific pair of scissors, uniquely programmed to recognize a different, distinct sequence in the DNA to make its cut. But how do these enzymes know exactly where to cut? To understand that, I've got to ask, do you remember what palindromes are? In everyday language, it's just a word that reads the exact same forwards and backwards. This is absolutely crucial for understanding our molecular scissors, because these restriction enzymes are literally patrolling the DNA specifically looking for palindromes. Now, what's really interesting about this slide is how we translate a normal word palindrome into a DNA palindrome. Take the word malleolum. Reads the same both ways, right? Well, in DNA, a palindrome is a sequence of base pairs that reads the same on the two strands when the orientation of reading is kept the same. Let's look at the specific target for an enzyme called EcoRI. On the top strand, reading from the 5' to 3' direction, it's GAATTC. Now look at the bottom strand, also reading 5' to 3'. It's exactly the same, GAATTC. The enzyme reads the DNA sequence forwards and backwards. Notice how the EcoRY enzyme finds its palindromic sequence, but it doesn't just chop it straight down the middle. Instead, it cuts the strand a little away from the center, but between the exact same two bases on the opposite strands. This leaves these single-stranded, overhanging stretches. These are those famous sticky ends Boyer discovered. We call them sticky because they naturally want to form hydrogen bonds with their complementary cut counterparts. This stickiness is what perfectly facilitates the DNA ligase, which, remember, acts as the final glue to join the molecules together. All right, on to section 4, sorting with gel electrophoresis. So we've just chopped our DNA into a bunch of tiny fragments. But how do we sort out the specific pieces we actually need? We use a pretty brilliant process called gel electrophoresis. You can think of it like a microscopic sieve powered by electricity. First, we place the DNA fragments into a matrix made of agarose, which is actually a natural polymer extracted from seaweeds. Second, we apply an electric field. Because DNA fragments are naturally negatively charged, they're forced to move toward the positive anode. The agarose gel acts just like a sieve, meaning the smaller the DNA fragment, the farther and faster it manages to move through that jelly-like matrix. Once everything is sorted by size, there is a slight catch. You cannot see pure DNA fragments with the naked eye. No way. So we have to stain the DNA with a compound called ethidium bromide and then blast it with UV radiation. When we do that, boom, these bright orange glowing bands of DNA suddenly become visible against the matrix. It's super cool. From there, we can literally cut the specific band we want right out of the agarose gel. And finally, section 5, vectors, our delivery vehicles. Now that we've got our perfectly cut and sorted piece of alien DNA, we need a way to get it into a new host organism so it can actually multiply. To do this, we use vectors, usually plasmids or bacteriophages. So the crucial point here is that for these vectors to successfully multiply our target DNA, they absolutely require a few specific components. First, they need an ORI, an origin of replication, to kickstart the multiplication. Second, they need selectable markers to help us identify which cells actually took up the DNA. And third, they need cloning sites, which are specific recognition spots for our restriction enzymes to open them up. And this brilliantly illustrates what we're talking about. Take a look at a real, heavily used E. coli cloning vector called PBR322. You can clearly spot the ORI right there, but pay close attention to those selectable markers. This vector carries specific genes for ampicillin resistance and tetracycline resistance. Notice how the cloning sites, like the target spots for enzymes such as BAM-HI, are located right inside those antibiotic resistance genes. Now, why on earth would we put the cloning site right in the middle of a resistance gene? Well, it sets up a really clever biological puzzle called insertional inactivation. If we successfully glue or ligate our alien DNA into the BAM-HI restriction site of the tetracycline resistance gene, we physically break that gene. The vector completely loses its tetracycline resistance. And here's the genius part. Breaking a resistance gene is actually our ultimate proof. It's delightfully ironic, but it allows us to easily differentiate our successful recombinants from the non-recombinants. It proves that our microscopic delivery vehicle successfully picked up the new DNA cargo. So, we've seen how restriction enzymes make the precise cuts, how electrophoresis sorts the pieces out, and how vectors deliver the final blueprint. The foundation built by scientists like Boyer and Cohen has completely transformed our world. As we continue to refine this microscopic factory to fundamentally alter the chemistry of genetic material, what groundbreaking therapeutics, vaccines, and biological solutions will we engineer next? The possibilities are entirely up to our imagination. Thank you so much for joining me on this explainer, and keep questioning the incredible mechanics of the living world.